name of my talk is the diurnal oscillation of leaf expansion is sensitive to VPD and atmospheric CO2. So, as we could hear in detail yesterday in the nice lecture by Professor Hetherington, and also today, the lecture by Professor McElwain, stomatas are very important or are key for the gas exchange processes at the leaf level. And they are very sensitive to environmental signals such as light, type of light, CO2 concentration, relative humidity, or well, VPD, and also AVA production. And in the context of climate change, where we are expecting a continuous increase of the CO2 concentration, maybe even doubling at the end of the century, this will have implications for the stomatal regulation and how this will change along the time. Also considering the optimization theory by Farquhar and Cowan that says that stomatas are capable of optimizing the relationship between carbon gain and water loss, a change in CO2 concentration is expected to alter stomatal conductance and also stomatal density. At the same time, stomatal conductance is important in the case of leaf growth, not only because the carbon gain will be defined both by the CO2 concentration but also by the stomatal regulation, but also because a different water loss or evapotranspiration due to stomatal decrease will impact leaf water potential and also leaf turgor. And according to Lockhart, we know that the rate cell enlargement is dependent on a wall extensibility factor, this M here in the equation, the reigning turgor at the points of cell extension, and a yield turgor. So this is especially interesting in the case of grasses because of two main reasons. The first one is that this has been shown that C3 grass, or well, in general, grasses are capable of better regulating stomatas. They have a more efficient stomatal control than other species, probably because of the architecture of their stomatas. And also because in the case of grasses, well, the growth happens in this enclosed environment, and it's mostly unidirectional. Therefore, following growth of leaves is rather simple in comparison with dicots. So with this scenario, we focus on the effects of CO2 and VPD on different parameters, including the leaf morphology and the leaf morphometry and cell morphometry, the effect on the leaf expansion rate at a daily average, but also considering the differences between dark and light growth, the stomatal conductance and proportionally the evapotranspiration, and the hydraulic relationships that could be having an effect on the leaf elongation rate. And we performed this analysis on perennial ryegrass, which is a C3 grass, pretty common in temperate pastures, and it's important especially for forage reasons. How we did this was with a control experiment in growth chambers, which we divided more or less in two parts. The first part having multiple chamber scale replicates to be sure about our results, and combining 3 CO2 levels and 2 VPD levels. The 3 CO2 levels will represent glacial minimums, the present day concentrations at what we could expect to have at the end of the century, and 2 VPD levels during the light period. These values, the low and high, represent temperatures of 20 degrees in both cases. Here is 75% humidity, and here 50% humidity. During the night, the VPD was kept constant. After a steady state was achieved in our canopies, we performed the morphometric analysis and also gas exchange measurements, both at the leaf level with this kind of system, and also at the canopy scale level. In both cases, we used infrared gas analysis techniques. And as you may see here, we grow these plants in individual tubes with quark sand, and we irrigated them with a slightly modified Hoagland solution. It had a slightly lower nitrogen concentration than the original one. We didn't want to give the plants excess nitrogen. And the second part of the experiment with the same kind of growth chambers was performed to obtain information about these hydraulic signals or the water status of the plants. And we analyzed the leaf water potential in mature leaves and also the osmotic potential in the leaf growth zones where actually the expansion of the cells is produced. Both at the end of the dark and at the light period. And then we approximately calculated turgor. And I say approximately because we measure osmotic at the leaf growth zones and the leaf water potential in the mature leaves. However, as it was shown by Martre with barley, I think, the difference in osmotic potential, in leaf water potential between at the leaf growth zones and at the mature leaves is relatively small. I'm talking about 0.1 megapascal. And if there is an error, it will be systematic throughout our treatments. In this case, we analyzed the extremes, so 200 ppm and 800 ppm. And coming to the results, the first result that struck us or we found rather surprising was that we didn't find almost no differences in leaf morphometry and cell morphometry regarding our more or less control, the 400 ppm treatment. And I say rather surprisingly because considering that the CO2 treatments especially were rather extreme, we would have expected some sort of CO2 fertilization, especially at the highest level, and maybe a reduction in these morphometric values for the 200 ppm. However, reviews like the one of Ainsworth in 2005 and a more recent one by Kimball with crops showed that this CO2 fertilization effect, even though it's constant in phase experiments, it may vary rather dramatically depending on covariant factors like nitrogen status, water status, or even season of growth. So for us, this was important thinking about how actually the biomass could increase in the future with grasses. According to these results, the leaf elongation rate, didn't change. It was unaffected by CO2, VPD, or their interaction. But what we observed was a rather high difference between the rates during the dark and during the light. This is also not so surprising because it has already been observed in grasses such as barley, rice, or also in Festuca arundinacea. But what we find very interesting is that the effect of CO2 and VPD was significant when considering the different periods separately. And even more interesting was the interaction between the light-dark regime and this effect. While in the light, CO2 increased leaf elongation rate and a lower VPD also, exactly the opposite effect was observed during the dark period. So coming to stomatal conductance, the results were more or less expected. Higher CO2 caused a decrease, a clear decrease in stomatal conductance, and also a lower VPD produced a higher conductance. However, it's remarkable how strong actually the regulation of stomatas is. While at 200 ppm, the difference between VPD levels is more than 100%, more than double. At 800 ppm, there is almost no more regulation, probably because they are also at lower levels. This repercuted in the evapotranspiration. So we observe more or less of a linear relationship, an inverse linear relationship between CO2 and evapotranspiration. But the VPD effect almost disappears and it was not significant. This has to do with the trade-off between higher conductance at a lower VPD level, but at the same, a lower leaf-to-air VPD, and in the end, this cuts out. And this effect on the evapotranspiration, these differences that we observed during light conditions, repercuted or had a correlationship with the leaf elongation rate during the light. So this shows us more or less that these differences had to do with some kind of hydraulic signaling. And the detail about these hydraulic relationships, we observed the expected the full bars are the dark conditions and the others are the light conditions. A more favorable water status during the night than in the light, associated with the stomatal closure in the absence of light, also turgor. And in the osmotic potential, the differences are rather small, what will suggest that the carbohydrate concentration or the solute concentration didn't differ too much between the different periods. Also to observe is that while in the night there were no difference in the water status, and this is logical considering that the VPD was constant, during the light there was a clear effect between the 800 ppm and the 200 ppm, what we associate with different transpiration rates. This becomes clear here in this graphic with almost a perfect inverse correlationship between the transpiration at the leaf level and the water potential, what will also indicate that the total hydraulic resistance was constant throughout our treatments, didn't change. And accordingly to this, also we observed that turgor had an effect on this leaf elongation rate. So until this point in the light, more or less it is clear that a hydraulic signal was controlling the expansion rates of the cell. However, this doesn't explain why there were so enormous differences in the leaf elongation rates during the dark, where the hydraulics were more or less the same. So what we think was happening is that we observed an stored growth effect. That means the leaf elongation rate was able to recover in the dark period after these changes in the hydraulics. This stored growth effect has been observed, for instance, by Procius and Bayer with algae, and it's shown that the cells are capable of expanding more than it will be expected without the previous drop in the hydraulic signals. And this is important for considering how actually the leaf growth processes could see in the future when CO2 will have an effect on the concentrations as also on transpiration effects. And finally, just looking at the summary, we didn't observe any difference among treatments in the leaf elongation rate at a diurnal scale, and also therefore on the morphometric values. But what we observed is a very clear and significant effect of CO2 and VPD in this diurnal oscillation. And also stomatal conductance, as expected from the literature and also from the theory, was strongly influenced both by CO2 and VPD, and this had an effect on evapotranspiration. And the differences caused by transpiration rates produce difference also in the leaf elongation rate in the presence of light. Additionally, we expect in the future for the plants to have a more favorable water status. It's also well known that the water use efficiency will increase with increasing CO2. I will say especially in the case of C3 species. And even though we didn't see differences in water status during the night, we observed tremendous changes considering the CO2 steps. And we associate this with an effect of a stored growth that is being observed then in the result and in the growth. So yes. And before finishing, I would like to thank my co-workers at the Grassland Group at the Technical University of Munich, my supervisor, the German Research Foundation for the support and the funding for our project, and of course New Phytologists for this nice conference, and you for your attention. And I will be happy to answer your questions.